Alright everyone, so now that we have jumped around and started blowing on our bromothymol solution in our beakers, let's go ahead and start doing our observation team. So the data that you will be seeing here, it will be different from yours, but this is just for you to use an example to follow. So for example, now that you did the blowing into the beaker, let's go ahead and do the observation table. So the observation is asking us the effect of the exercise of the rate of cellular respiration, right? So when we're breathing in and out, our bodies, our cells are doing cellular respiration. So you see the bar here, you see the little table, we see that it says time length of activity, and we got the time for color change in seconds, right? So when we talk about 0 minute exercise is when you all did the blowing into the bromothymol, so blue solution without exercising. This is where we're going to put our data. And for example, mine was, it took 12 seconds to change color, right? So when I did not exercise and I did the experiment, it took 12 seconds for the bromothymol or the solution to change color. Now, when we talk about the 1 minute exercise, we're talking about after we did our jumping jacks or running in place for 1 minute, and for that I got 6 seconds. Remember, my data is probably a little bit different than yours. Now, if we go here where it says prediction, question 3, we're not doing this just yet. We're gonna do this once we jump into our to our question 3. All right, so after you have done, you did your observation table and you collected your data. Let's go ahead and go into the analyze and apply. So the analyze and apply is asking us to make a graph of our results. So determine if the graph should be a line graph or a bar graph, and then carefully decide which variables belong on the x-axis and the y-axis. Remember to provide a title and to label the x and y-axis, right? So for my example here, I have on my x-axis time length of the activity, and then I have minutes on my right here. So I need to make sure that I include the measurements, which is time length of the activity, which is on my x-axis, right? But we also need to include the minutes if we're doing our bar graph right here. And for our y-axis, we're doing the time for color change. This is when you blew into your beaker to see how long it took for the solution to change color, right? So we got to make sure that we put our measurements, which is time for color change, and we also need to include the seconds, right? Because we measured the time change in seconds, and we need to include our y-axis. Also on our y-axis, I included the numbers 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You can go ahead and do every 5. You can go ahead and put, for example, I did for every, here I put all the numbers, but you can just go to 5, and then you can go to 10, and remember each little line would be 1 second. All right, so now how I graph my time graph, my graph, right? So right here we can see that at 0 minutes, this is the time, the length of the activity. That's when we did no activity. When I blew into the beaker of the solution, I can see right here that it took 12 seconds for it to change, right? So when I had no activity, like jumping jacks or running in place, I saw that it took 12 seconds for my solutions to change. So it changed color after the 12 seconds. Now on this section, what you're going to do is that after your 1 minute of jumping jacks or after 1 minute of running in place, you will see that in my example, when I did 1 minute, it took 6 seconds. So if we go here, it took 6 seconds for my solution to change color. So you will get a graph like this. Remember, it might be different than mine. So go ahead and start graphing your solution change, your time length of activity, and your time length for color changing section on your bar graph. All right, so now that we went ahead and did our bar graph, we can actually answer question number 2. So now question number 2 is asking us, compare the time it took for the bromothymol blue solution to change color before exercising and after exercise, right? So we noticed that when there was no exercise, you can look back to your bar graph, when we had no exercise, 0 minutes of exercise, it took 12 seconds for it to change colors, right? So we can go ahead and put that on our question 2, our answer. We saw that when before the exercise, it took 12 seconds, right, to change color. So let's go ahead and be specific. 12 seconds to change colors. And now it's asking us the color change after the exercise. So we can go ahead and put that after exercise. It took how many seconds for me? Nice, 6 seconds to change color, right? Nice. So now it's asking us to explain why there was a difference. So go ahead and raise your hand if you know why there was a difference. Let's go back into our cellular respiration, into our equation. See, that may give you an idea of why there was a difference. Nice, yes. So there was a difference because when we did no exercise, we saw that there was only a change. It took a whole 12 seconds for it to change colors, right? But after we exercise for a full minute, when we're doing our jumping jacks and we're doing our running in place, we saw that it took only 6 seconds to change colors, right? And if we go all the way back to the beginning of the page, it tells us right here that when carbon dioxide is dissolved in water, it creates carbonic acid. This reaction occurs as rainwater absorbs carbon dioxide, right? So we can see that after we were running in place for a full minute, we were exhaling more, yes, carbon dioxide. There we go. So that's the reason why it took less time to change, right? So the more we exercise, the more carbon dioxide that we exhale. And remember, this happens when we're doing cellular respiration. We get our sugar, which is our glucose. We breathe in oxygen when we're inhaling. And then when that happens, oxygen and glucose turn into carbon dioxide, which we exhale, the water, which is what the cells absorb, and ATP, the energy, which is what it took us to actually do our jumping jacks and running in place. So now, using your graph, we're going to go number 3. Using your graph, predict the time of color change if the experimenter did exercise for 2 minutes, right? So let's go ahead and look back at our time graph. So let's go ahead and start predicting. So we see that after no exercise, we took 12 seconds, right? But after 1 minute, it took 6 seconds. So what would happen if I did this for a full 2 minutes? Let's go ahead and look at the pattern, right? So 0 minutes of no exercise, 12 seconds to change color, and 1 minute of exercise, it took 6 seconds to change colors. Okay, now let's go ahead and go back to observation table, right? So for our prediction, let's look at our table. Can we see a pattern? Nice, exactly, yeah. So we're seeing that after 0 minutes of exercise, it took 12 seconds. After 1 minute of exercise, it took 6 seconds. Then maybe, probably, after 2 minutes of exercises, since we're doing a prediction, it would probably take, nice, yeah, 3 seconds. Oh, let me do a check that for you. There we go, 6 seconds, 3 seconds. So we're seeing that it's every time we do a minute, it's cutting the time for it to change color to 12 seconds. We can go ahead and actually go back and start graphing this. So when we're graphing this is where we're skipping a line, and then we look at our x-axis. Here we're doing 2 minutes. We need to make sure that we specify the measurement, in this case 2 minutes, and we're going to go into our x-axis 3 seconds. 1, 2, 3, which is go here. So x-axis 2 minutes, and then we go up 3 seconds. You can see that we predicted that for after 2 minutes of exercising, it would take 23 seconds for the solution to change colors. So now we can actually go ahead and answer question number 3. So now using your graph, predict the time color change if you experience the exercise for 2 minutes, right? So you can put after 2 minutes. So after 2 minutes, oh, spelling, here we go, 2 minutes. So after 2 minutes, my prediction is that it will take 3 seconds for my solution to change colors. Remember, so this is our prediction. And remember, you're probably going to get a different number than mine, and that's okay. So now, if we go to number 4, in this investigation, we measured the amount of carbon dioxide produced to find the rate of cellular respiration. And we did, right? We did this. We can look back at our table all the way to the top, and we can see that for 0 minutes, after no exercise, it took 12 seconds to change colors. And then we could say the same thing for 1 minute. So after 1 minute, we saw that the color change was at 6 seconds, and after 2 minutes of exercise, it only took 3 seconds. Right, so this is our rate. So no exercise, 12 seconds. 1 minute of exercise equals 6 seconds of when it took to change colors. And after 2 minutes of exercise, it took 3 seconds to change colors. So now, what else could we measure to find out the rate of cellular respiration? So for this, what else we could measure to find out the rate of cellular respiration? Let's go ahead and look at our formula, right? So look at our formula, see what else we can use to actually measure the rate of cellular respiration. So we can see here, we already used carbon dioxide, right? So what else can you measure? Yes, great. So we can also measure the oxygen, right? In this solution, in this experiment, we'll be able to do the oxygen, but there are apparatus or there's technology already that we can use to actually measure the intake of oxygen, right? So when we see our formula, we see that when we inhale and we take the glucose in our body, we create carbon dioxide, water, and ATP. So that means that if when we're producing more carbon dioxide, what else do we need to take in? We will need to take a lot of what? Nice, oxygen. So we need to take a lot of oxygen and a lot of glucose. Our body will have to take all the glucose so we could go ahead and turn it into carbon dioxide. So you see that all this formula, they're related, right? They're related. So if you want to produce more carbon dioxide like we did in this experiment, we have to have more oxygen and we need to have more glucose. That's the way we can go ahead and see that more carbon dioxide will be produced. So what else can we measure to find out the rate of cellular respiration? If you put oxygen, that's correct. We could measure the amount of oxygen taken in when we breathe. And you actually saw this while you guys were doing your experiment, right? You were seeing that the more you were running, the more oxygen you were taking in, right? So you were breathing in and breathing out faster because your body was trying to produce that energy. So we go back to our cellular respiration. We could see that when we were breathing in a lot, we were taking in a lot of oxygen. Our respiration was up and down, up and down. Our chest was going up and down, up and down. We could see that we were taking a lot of oxygen and we were producing a lot of carbon dioxide because we needed that energy right here. We needed this energy to actually keep our legs moving and our hands moving when doing jumping jacks or we were running in place. So you could also measure the amount of oxygen taken in when we breathe. Now, number 5 is asking us, explain why it is important to exhale completely when doing the exercise. So when we were breathing into our test tube, we were breathing out the carbon dioxide. So why do you think it was important to completely breathe out in this exercise? Yes, of course. Nice. So it was important to exhale completely because remember when you're breathing out and then you're exhaling, you need to get all those molecules of carbon dioxide out of your body, right? So it wouldn't mess up your experiment. Also, if for a safety reason, we had to exhale completely and then take our mouth out of the straw to actually breathe in the oxygen and then put our mouth back in the straw to go ahead and put the carbon dioxide back in the solution. But it was most important because we needed to breathe out every particle of carbon dioxide that our body was making. If not, it would take more time for the solution to actually change colors, right? So you can put, it is important to exhale completely so that all the molecules of carbon dioxide, CO2, CO2. So it was important to exhale completely so that all the molecules of CO2 would go in the solution. In the solution to change colors. For the solution to change colors, let's go ahead and change that. So yes, it was important to exhale completely so all the molecules of carbon dioxide would go in the solution and for the solution to change colors. That was the reason, right? Because if we were breathing in all the oxygen and we're only exhaling halfway, all the molecules are not going to dissolve in our solution. So it would mess up our data and it would actually take longer for our solution to actually change colors.